A huge debate in the longevity community centers on a single question: what truly causes aging? Entire research programs, biotech companies, and philosophical worldviews depend on the answer. And within this debate, two major theories dominate: damage accumulation and programmed aging. The first proposes that aging is the gradual consequence of wear and tear at the molecular level. Over a lifetime, our cells experience DNA mutations, oxidative stress, protein misfolding, mitochondrial decline, and a variety of biochemical insults. Slowly and steadily, these forms of damage build up. Eventually, the total burden overwhelms the body's ability to repair itself, and the organism breaks down. The second theory suggests something different. Programmed aging argues that organisms evolved to deteriorate after a certain point because doing so once granted an evolutionary advantage. In this view, aging is not a passive collapse, but an active developmental phase, shaped and orchestrated by biological pathways. In other words, death is written into our operating system. Dying is quite literally in our genes. At first glance, this distinction might appear academic because the outcome is identical. We grow old and we die. But understanding which mechanism is correct may be one of the most important scientific questions of our time. If aging is caused by damage, then we must focus on repair and maintenance. If aging is programmed, then we can focus on reprogramming the body's internal instructions. Each path leads to a very different future for medicine, biotechnology, and human longevity. Determining the root cause will shape whether we treat aging like rust on a machine or like a software script that can be rewritten. So let's start with the damage accumulation side of the debate. This is the view that seems to be held more traditionally in biology. As we live, our bodies slowly accumulate wear and tear, not metaphorically but literally. Cellular damage is measurable and it increases with age in ways that are consistent. When researchers sequence individual cells from younger and older people, they notice a difference. Older cells carry more mutations, not enough to cause immediate disease in most cases, but enough to subtly interfere with the precision of cellular function. Even in tissues that barely divide, like neurons, this slow creep of DNA damage is happening. It is a quiet, steady march of entropy. First, there is telomere shortening. Every time a cell divides, the protective caps on the ends of our chromosomes get just a little bit shorter. Over time, they hit a critical length and trigger the cell to stop dividing altogether. Over decades, this gradually erodes our regenerative capacity. Cuts heal more slowly, tissues bounce back less, and stem cell pools start to thin out. Proteins show similar changes. As we age, the systems that fold and repair these proteins become less efficient. Misfolded proteins appear more frequently, and the machinery that breaks them down struggles to keep up with demand. A young cell might run like a well-maintained machine, but an old one is held together with duct tape and glue, metaphorically speaking. We see this most dramatically in neurodegenerative diseases. Take Alzheimer's. Yeah, there is an early onset form, but it's quite rare, roughly 5% of all Alzheimer's cases. The overwhelming majority happen in old age, which fits the damage accumulation model. After decades of oxidative stress, protein misfolding, mitochondrial dysfunction, and DNA damage, the brain eventually reaches a point where its ability to compensate is overwhelmed. A buildup of amyloid beta, a protein fragment derived from amyloid precursor protein, forms sticky plaques outside neurons. Inside these neurons, another protein called tau becomes hyperphosphorylated, detaching from microtubules and forming tangles. Together, These plaques and tangles disrupt brain cell function and lead to the memory loss and cognitive decline we see in Alzheimer's. On a slightly tangential note, this is partly why some people in the longevity community argue that aging should be classified as a disease. If the biological deterioration that leads to Alzheimer's, heart disease, frailty, cognitive decline, all emerge from the same underlying processes, then perhaps we should treat aging itself as the pathology. If we begin to view aging as a disease, we open the door to interventions and treatments just like any other medical condition. This perspective could allow regulatory bodies such as the FDA to approve therapies specifically designed to slow or reverse aging, transforming how we approach both longevity and health span. Now, the disposable soma theory tries to answer a simple but fundamental question. If aging is so harmful to us, then why didn't evolution design bodies that maintained themselves better? The answer to that question lies in how evolution actually works. Natural selection is not trying to create the longest lived organism or the most durable body possible. It only cares, in so much it has a thought process, about one thing. Can you survive long enough to successfully reproduce and pass on your genes? Everything beyond that point matters far less, because once you have produced offspring, your genetic job, from nature's perspective, is complete. In early human history, life was harsh. Predators, infections, accidents, environmental dangers. Very few individuals ever had the chance to make it to old age. That means that there was very little selective pressure to fine-tune biological systems for long-term maintenance. I mean, what would be the point of living to 200 if almost half your generation never made it past adolescence, failed not by age, but by disease, accidents, violence, the harshness of nature? In that situation, you have way bigger fish to fry than aging. Evolution invested its limited resources into rapid growth, fertility, wound healing during youth, and the traits that directly increases reproductive success. Those investments gave a strong early life advantage, even if they came with hidden long-term costs. Our bodies are built to be maintained, but only to the degree that makes sense in the environment where we evolved. Maintenance systems were designed to be good enough, not perfect. Over decades, small inefficiencies accumulate. Damage slips through repair mechanisms. What we experience as aging may simply be the long-term outcome of a body optimized for survival and reproduction in the short term at the expense of indefinite longevity. And this pattern isn't unique to humans. When you look across species, you see the same logic everywhere. Animals that face high predation pressure tend to mature quickly, reproduce early and often, and then age rapidly. Their evolution strategy is to live fast, reproduce fast. On the other hand, species with safer ecological niches, animals with fewer natural predators or more stable environments, tend to evolve slower aging, longer lifespans, and more robust maintenance systems. Tortoises, whales, naked mole rats, parrots, they aren't immortal, but evolution has clearly invested more resources into keeping their bodies intact. All of them have long lives, comparatively speaking. And we see the opposite end of this spectrum play out at the microbial level. Bacteria are some of the most successful organisms on the planet, and part of their success comes from evolution working fast and efficiently. Look at antibiotic resistance. Most antibiotics humans have developed have been around for less than a century, less than 100 years, barely a blink in evolutionary terms. Yet bacteria have already evolved countless mechanisms to resist them. They don't need to maintain themselves forever. They only need to survive long enough to reproduce. Bacteria are a living demonstration that evolution prioritizes reproductive success over longevity, and it does so with ruthless efficiency. If we want to see what happens when evolution does not prioritize adaptability, we need only look at the dinosaurs. When 13-meter-long lizards with teeth the size of your arm roamed the earth, mammals struggled to survive. We were confined to small ecological niches, keeping ourselves small to avoid predation. But when the earth began to change, dinosaurs could not adapt quickly enough to the shifting environment. Mammals, poised to take their place, inherited the earth, leaving a vital lesson for evolution. Long lives slow down the pace of change, stretch the gap between generations too far, and species evolve too slowly to keep up with the changing world. Taken together, all of this creates a compelling picture of aging as a slow breakdown, a gradual unraveling that begins almost imperceptibly in youth and accelerates as the burden of damage grows. Not a switch being flipped, but a long and steady fade. So if damage accumulation and the disposable soma logic were the full story, then why does another theory keep being considered? And perhaps more importantly for this video, why do I believe that this other theory better explains what we see? At the core of both views is the survival of genes, and ultimately the survival of the soma long enough to reproduce. But I don't think damage buildup tells the whole story. Ironically, when people point to other long-lived animals whose internal mechanisms simply work better, they're kind of pointing to evidence for programmed longevity. Those species are in fact built different because their biological code was tuned to live longer. Humans undergo menopause, or rather women do. I know that's apparently a controversial statement, but women undergo menopause. Women's window of fertility is relatively short and tightly defined. If aging was purely about accumulating damage over a lifetime, you'd expect a lot more variation in the age at which menopause hits. You might expect someone with great genes and a pristine lifestyle to have menopause much later than someone whose health is compromised. You know, that one who smokes and drinks, are overweight, they're stressed, they do a bunch of drugs. But in reality, though lifestyle changes can shift things a few years, the range remains surprisingly narrow compared to the theoretical variation. And this consistency suggests something more clock-like, a scheduled shutdown. If evolution can maintain reproductive fitness for, say, 20 years or more, why does the system then undergo such a rapid and uniform decline? The body has already solved the problem of maintenance for those years, yet once reproduction ends, a cascade begins. I mean, one could invoke selection shadow, that after reproductive age, natural selection exerts weak pressures so errors can slip through, but that feels like a much weaker and much more convoluted explanation than programmed decline. As we know from Occam's razor, often the simplest solution is the correct one. Here's a question for you. Out of all of the animals in our vast animal kingdom, how many do you think undergo menopause? All of them? Most of them, bar a few rare exceptions? More than half, surely. The correct answer is 6, to the best of our knowledge at least. See, the rarity of menopause across the animal kingdom just adds further weight to this. There are over 5,000 mammal species, yet we have only observed 6 which undergo true menopause. That's humans plus 5 different species of toothed whales of all things. The killer whale, the short-finned pilot whale, the beluga whale, narwhals, and false killer whales. I mean, we may not know all the answers, admittedly. There is some debate on Asian elephants. There was a study that found that about 13% experienced a true post-reproductive phase, but that's still only 13%. Most of them don't exhibit it. And even in chimpanzees, our closest relatives, it's generally not observed. Again, there is some evidence, and in fact a very recent study from 2023, but that transition is not species-wide. It's the minority, with most chimps remaining reproductively active well into their 40s and 50s, which is exceedingly old for a chimp. So let's be charitable and include Asian elephants and chimpanzees in this discussion, even though the results are inconclusive at best. That's 8 species. 8 species out of 5,000 mammals. That equates to 0.16%. And if you widen the lens to all animal species, birds, reptiles, fish, invertebrate, the proportion is vanishingly small, less than 0.01%. The fact of the matter is that the vast majority of animals reproduce until near death. Menopause is not a typical byproduct of living a long life. That rarity suggests menopause did not emerge by accident. It likely evolved only under very specific ecological and social conditions. In these species, and that includes humans, the older non-reproducing females provide valuable roles. They're caregivers, they provide social leadership, they transfer knowledge, they boost the survival chance of their descendants. And there is so much literature showing that evolution has evolved to have altruistic behaviors. Even if we don't reproduce ourselves, we can look after our nieces, our nephews, because from a biological perspective, they share enough of our DNA that helping them to survive is still fulfilling our biological imperative. It's still allowing the soma to pass on. And these altruistic and caregiving traits is what helped us to build the society we have today. The good parts, at least. I know there's plenty of bad, but that's a topic for a later video. When you look at evolution broadly, from whales to humans, the fact that menopause and the long post-reproductive lifespan it enables has evolved independently in so few lineages suggests it is an evolved, regulated strategy. It is more plausible that what we call aging in humans is not just a slow breakdown, but part of a programmed process. Let's look at the biochemistry, the pathways that make you grow, IGF-1, growth hormones, mTOR. They help us reach our adult size, they kickstart puberty and hit our reproductive peak. But when those pathways are overactive or when they go unchecked, they also accelerate aging. Take progeria, or Hutchinson-Gilford-Progeria syndrome if you want to be pedantic. Kids with this condition carry a mutation in the LMNA gene that makes their cells, well, act like they're trying to be on a summoning salt video. They speedrun it, they fast forward, they age rapidly. Their growth and development occurs, but the same cellular machinery that supports life also breaks it down quickly. They experience hair loss, cardiovascular disease, all the other hallmarks of aging decades earlier than normal. And let's take it further and look at progeria's cousin, Werner syndrome, which is often called adult onset progeria. Patients develop normal puberty, but then in their 20s and 30s they start showing premature aging signs, graying hair, skin thinning, cataracts, metabolic decline. Does this not suggest that something has gone wrong in their programming? The natural plan set out for them has been fast-tracked, but that plan was there all along. Surely if it was just an accumulation of damage, we wouldn't expect to see uniform decline in so many systems. If something has snuck through and damaged the genes, you would expect a specific problem, not problems everywhere. Even in more subtle ways, the hallmarks of aging, things like cellular senescence, map almost perfectly onto normal life stages. You have your growth, your reproductive peak, and then the eventual decline. It's as if our biology is using the same switches to build us up and later to wind us down. If aging were just random damage piling up, you'd expect it to be messy, unpredictable, like a car or a building falling apart without warning. But our bodies have clocks, precise, internal timers that tick along whether we like it or not. Scientists like Steve Horvath have shown that chemical marks on our DNA, the way genes are turned on and off, change at consistent rates. These clocks, like the Horvath clock or Grim age, can even predict chronological age with accuracy, astonishing accuracy. It's like our cells carry their own countdown timers. Even our immune system has a clock, the T-cell repertoire, basically the variety of immune cells ready to fight diseases. These slowly contract over time. You aren't randomly losing these cells. The decline follows a schedule. The thymus, where your T-cells are trained, they shrink on a precise timetable. This process, known as thymic involution, is not messy wear and tear. It's a programmed shrinking that starts in childhood and continues throughout life. Across the tree of life, death isn't always random. In some species, it's literally programmed to happen once reproduction is complete. Nature has a blueprint for it, and it's almost always tied to reproduction. Take salmon. After a single spawning season, they die, their bodies fall apart. They die immediately after reproducing, which provides nutrients not just to the ecosystem, but it also reduces competition for their offspring and makes room for the next generation. Their death is not an accident, it's a strategy. And the same occurs when we look at the smaller cousins, the insects. The lifespan of a worker bee is drastically shorter than that of a queen. The queen lives for years, constantly reproducing, while the workers have much shorter task-driven lives. These lifespans aren't random, they're programmed to optimize colony survival. Early death of workers benefits the colony as a whole, preventing overcrowding and ensuring resources are directed toward reproduction. From an evolutionary perspective, rapid generational turnover has clear benefits. It accelerates adaptation, it ensures genetic diversity, and it reduces competition for resources. There's even a hypothesis that aging might help keep pathogens in check. If hosts die after a certain age, microbes have to constantly adjust, making the host a moving target, so to speak. The key point here is that there is a precedent in nature for organisms to have programmed death. Salmon, bees, countless other smaller examples. If death can be programmed in other species, why would humans be any different? The only change is the way in which it manifests. Older individuals provide benefits to their species and communities beyond just their reproductive capabilities. I have no doubt that many of you were looked after by your grandparents at some point because it freed up your parents and allowed them to work and provide. And if this programmed theory is correct, then it's good news. If aging were just a random pile-up of cellular damage, then reversing it would be extremely difficult, far more difficult than the alternative. You would need to tackle every hallmark of aging, individually targeting all of the different systems that started to decline. And people are attempting that through things such as SENS, strategies for engineered negligent cell senescence, pioneered by scientists like Aubrey de Grey. But as much as I respect the man and the work he has done over the last few decades, I do not think his approach is the best way about it. Scar tissue, broken DNA, misfolded proteins, these things don't come with an undo button. Fixing all of that would be a monumental undertaking. But what if we didn't have to? What if we could go straight to the source and tackle the root of the problem? Scientists have been able to partially reprogram cells to a younger state using techniques that reset epigenetic markers, the chemical switches that control gene expression. Skin cells, for example, can be reverted to a more youthful state without erasing their identity. And yes, to date it's only been done in vitro, we haven't been able to do it on a person, but the proof of concept is there. And organs in mice, I mean, they've been rejuvenated in similar experiments, improving tissue function and extending lifespan. So what does this tell us? Well, it suggests it's information based, stored in epigenetic programs and regulatory networks that can, at least in principle, be rewritten. If we can tweak the software of the cell, we can reset biological age. This is an important conversation we need to be having, because if aging is programmed, then reversing aging isn't about fixing every little thing that breaks. It's just about rewriting the code.